Imagine fighting an enemy that, literally changes its armor every single time you change your weapon. Like you bring fire, it becomes fireproof.
Right, or you freeze it and suddenly it just thrives in the cold.
Exactly.
Yeah.
And that is glioblastoma, or GBM. It is literally the most common and sadly the most devastatingly lethal adult brain cancer out there.
It really is. I mean, if you look at the statistics, you are looking at a median overall survival rate of just 15 months.
Which is just wild to think about. But here is the detail that really highlights the scale of this failure. There have been absolutely no new targeted therapies approved for glioblastoma since the drug temozolomide was introduced.
Yeah.
And that was all the way back in 2005.
Yeah, 2005, which represents an immense layer of frustration across the board, for patients, obviously, but also for clinicians. I mean, the scientific community has poured billions of dollars into fighting this disease, and yet the tumor consistently outmaneuvers us.
So today, we're taking a deep dive into a brand new paper from Nature Communications. This just came out in January 2026, and it completely reframes how we look at this problem.
It really does. Our mission today is to map out exactly why GBM is so impossibly hard to eradicate.
Right, and then we are going to explore this really novel computational framework called scFOC. It's essentially a mathematical blueprint that, it might actually give us the predictive power to finally trap this cancer.
But to understand why scFOC is so revolutionary, we first need to confront the core biological hurdle here. The paper identifies this main villain as intratumor heterogeneity and plasticity.
Okay, so let's translate that. Rather than thinking of a tumor as a monolith, like just a solid lump of identical clones, we need to look at it more like a piece of highly sophisticated polymorphic malware.
I love that analogy. You don't just have one piece of bad code, you have distinct subroutines.
Right. And in a glioblastoma tumor, these subroutines are distinct cellular states. And researchers have been using single-cell RNA sequencing to essentially reverse engineer this code base.
Exactly. And let's clarify that real quick. Single-cell RNA sequencing lets us look beyond the bulk tissue. Instead of grinding up a whole tumor and getting an average, we analyze the gene expression profile of every individual cell.
So you're zooming all the way in.
Right. And when they did this with glioblastoma, the transcriptomes revealed something really insidious. These GBM cells are basically hijacking normal neurodevelopmental pathways. They mimic the brain's natural cellular infrastructure, but in a totally chaotic, malignant way.
So they isolate 4 primary cancer states within the tumor.
Yeah.
Right. Let's break down the biology of these 4 states, starting with the astrocyte-like or AC-like cells.
Sure. So in healthy neurobiology, astrocytes are critical support cells. They maintain the blood-brain barrier. But in GBM, these AC-like cells are highly deregulated. Their defining signature is the massive overexpression of a gene called EGFR.
The epidermal growth factor receptor, right?
Spot on.
So they just have this hyperactive receptor constantly signaling the cell to divide and grow, completely ignoring the body's normal stop signals.
Precisely. Then, shifting gears, you have the mesenchymal-like cells, or the MES-like state.
And mesenchymal transitions usually have to do with tissue repair.
Right.
And mobility.
They do, and that's exactly what makes the MES-like state so dangerous. These cells are associated with tissue injury responses and immune evasion. They are highly invasive.
So they are the ones infiltrating deep into surrounding healthy brain tissue.
Yes. And their primary molecular calling card is the overexpression of CD44. That's a cell surface glycoprotein involved in cell adhesion and migration.
Okay, got it. So AC-like cells drive rapid growth via EGFR, and the MES-like cells drive invasion via CD44. What about the final 2 states?
The final 2 are closely related. You have the neural-progenitor-like, or NPC-like, and oligodendrocyte-progenitor-like, OPC-like. They function like the chaotic stem cells of the tumor ecosystem.
Meaning they are locked in a perpetual state of division.
Right. Both the MPC and OPC-like cells overexpress genes like CDK4, which is a major driver of the cell cycle, and SOX4, which is a critical transcription factor in embryonic development. They are constantly supplying the tumor with new raw material.
Okay, so let me do a quick recap here to make sure we're locking this in. Number 1, GBM tumors are heterogeneous, meaning they are made of multiple cell states. Number 2, those 4 distinct states are AC-like, MES-like, NPC-like, and OPC-like.
Perfect summary.
But here's my question. If we know the 4 types, Why can't we just mix 4 drugs together, like a heavy-duty cocktail targeting EGFR, CD44, CDK4, and SOX4 simultaneously? Just nuke all the subroutines at once.
Well, that would absolutely work if these cells were static. But it ignores the second half of our villain's description, which is plasticity.
Meaning they are fluid.
Highly fluid. When you attack them with standard of care treatments, you apply selective pressure. The cells sense the environmental toxicity and literally undergo a phenotypic shift. Yeah, so if you deploy a drug that brilliantly targets the NPC-like cells, those cells will alter their gene expression, downregulate CDK4, upregulate CD44, and just morph into MES-like cells to survive.
So the malware literally rewrites its own code in real time in response to our antivirus software. The target doesn't just move, it fundamentally changes its identity.
Exactly, and that perfectly explains why targeted therapies have been failing for 20 years. The moment you lock onto a vulnerability, the tumor abandons it.
Which brings us to the third bullet point of our recap. These cells can shapeshift to evade treatments. So reacting to them after the fact is a losing battle. We need to predict their moves.
Which is exactly where the scFOCAL framework comes in.
Okay, let's get into scFOCAL. Single-cell framework for Amyx connectivity and analysis via L1000 pounds. It's a very serious acronym, but the underlying mechanism is fascinating.
It really is. It basically operates as a massive subtraction engine. First, it takes single-cell RNA sequencing from a patient's tumor and isolates the cancerous cells.
And it doesn't just look at the cancer in a vacuum. Right. It compares those cancerous cells to normal cells.
Right. Non-neoplastic cells like T cells and myeloid cells in the tumor microenvironment, it establishes a baseline.
So by subtracting the normal gene expression from the malignant gene expression, it isolates the exact genetic delta. It identifies precisely which genes the cancer has pathologically upregulated.
And which vital tumor suppressor genes it has downregulated. This delta is what the paper calls the disease signature for that specific cell state.
Okay, so once you have that specific disease signature, you need a way to counter it. And this is where the framework taps into the NIH LINCS L1000 pound database. Let's explain what that is.
The L1000 pounds is a massive catalog of transcriptomic data. Researchers took human cell lines, exposed them to thousands of different small molecule drugs, and measured exactly how each drug altered the expression of roughly 1,000 landmark genes.
So we know exactly what these drugs do to genetic expression. I really like thinking about this using a physics analogy like active noise-canceling headphones.
Oh, I like that. How so?
Well, the microphone on the headphones listens to the ambient frequency of, say, an airplane engine. To create silence, the speaker doesn't block the sound, it produces the exact inverted sound wave. The peaks of the engine sound wave meet the troughs of the headphones wave, they collide, and they cancel each other out.
That translates beautifully to transcriptomics. If a specific cancer cell thrives by upregulating genes X, Y, and Z, scFOCAL searches the L1000 town database for a drug that naturally downregulates those exact same genes.
It creates the exact opposite sound wave, and the researchers call this discordance, right, or disease signature reversal.
Exactly. If the drug signature is highly discordant with the cell's signature, the cell will be highly sensitive to the drug. And scFOCAL calculates this at the single-cell resolution.
Okay, time for another quick mini recap. So 1, scFOCAL creates a disease signature for each specific cell. 2, it searches this massive L1000 pound database of drug signatures. And 3, it looks for discordance to find the perfect molecular match to kill that specific cell.
Perfectly stated.
But, you know, a computational simulation running on a database is one thing. Living biology is messy. Did this actually work in a living brain?
It did. They set up their first major case study around a drug called alisertib.
Alisertib, that is an aurora kinase inhibitor, right?
Right. Aurora kinases are essential enzymes for cellular division. They help pull chromosomes apart. Now, historically, alisertib showed incredible promise in preclinical models, halting division and shrinking tumors.
But the tumors always grew back.
Always. And the mechanism of that resistance was a bit of a black box, so they used scFOCAL to illuminate it. The algorithm made a bold prediction. Alisertib would be highly discordant with and therefore kill the NPC-like cells, but it would be completely ineffective against the MES-like cells.
So to test this, they utilized an orthotopic xenograft mouse model. Let's unpack that term quickly.
Basically, they took molecularly classical glioblastoma cells, the GBM22 line, and implanted them directly into the brains of mice. So the tumor grows in its native microenvironment.
Right. And then they treated them with alisertib. What happened?
The results exactly matched the computational prediction. The NBC-like subpopulation was drastically depleted, but the proportion of MES-like cells absolutely skyrocketed.
Okay, wait, here's where it gets really interesting to me. If our treatment wipes out the NPC-like cells, but the tumor just uses its plasticity to shift into that highly invasive MES-like state, haven't we just backed ourselves into a corner? Like, haven't we actively made the tumor worse and completely resistant?
In traditional oncology, yes, that's a clinical failure. The tumor outsmarted the therapy. But with scFOCAL, this is actually our greatest advantage.
How so?
Because it's no longer a reactive guessing game. It is evolutionary steering. We didn't just stumble into a resistant tumor. We mathematically forced the tumor into a highly predictable state.
Because we can predict what it will turn into, we can have a second trap waiting at the finish line.
Precisely. By forcing the tumor to adopt the MES-like state to survive the alisertib, we radically alter its dependencies. scFOCAL showed that those new MES-like cells suddenly exhibit severe vulnerabilities to entirely different classes of drugs, like topoisomerase inhibitors or serotonin receptor agonists.
So we use the first drug to push the tumor off balance, and the second to exploit the new vulnerability. Let's do recap number 3. 1, alisertib kills NPC-like cells, but leaves MES-like cells alive. 2, the mouse models proved this prediction flawlessly. And 3, by knowing what survives, we can predict the tumor's next vulnerability.
Which naturally transitions to the holy grail of this research, which is rational drug design, building synergistic combinations from scratch.
Okay, let's look at the ultimate tag team they designed. What was the first punch?
They started with a drug called CT-179. This is a targeted inhibitor of OLEAD2, which is a critical dependency for maintaining the tumor's hierarchy.
So scFOCAL predicts that inhibiting OLE2 will effectively target the OPC-like cells, right?
It does, but staying true to its plastic nature, the model forecasts that a resistant population will survive, and they will overwhelmingly morph into the AC-like state.
And we know the surviving EC-like cells rely heavily on the overexpression of EGFR. So what was the second punch?
scFOCAL scanned the database for the perfect molecular countermeasure to an EGFR-driven survival state. It identified a specialized drug called Depatux-M, or ABT414.
Depatux-M is an antibody-drug conjugate, or ADC, and the mechanism of an ADC is incredibly elegant. Let's break that down.
Absolutely. ADCs are designed to solve the toxicity problem of traditional chemo by tethering a highly potent poison directly to a tracking system. It uses a monoclonal antibody that binds exclusively to the EGFR protein.
So that antibody acts as a homing beacon, constantly scanning for cells overexpressing EGFR. And attached to it is the cytotoxic payload, right?
Yes. In the case of Depatux-M, the payload is MMAF, a potent tubulin inhibitor.
So the antibody binds to the EGFR on the surface of the AC-like cell. The cell naturally internalizes the receptor, dragging the whole ADC inside.
Exactly. And once inside the acidic environment of the lysosome, the chemical linker degrades, releasing the MMAF payload directly into the cell. It disrupts the microtubule network, and the cell undergoes apoptosis. It dies.
So if we pull back and look at the entire trap, you hit the tumor with CT-179, it wipes out the OPC-like cells, the tumor mechanics relies on plasticity to survive, and forces its remaining cells into the AC-like state, aggressively upregulating EGFR.
And that very act of upregulating EGFR makes them the perfect illuminated targets for Depatux-M, which completely decimates the surviving population.
And when they tested this in vivo in mice, the synergy was profound, right? It significantly attenuated tumor growth and drastically extended survival.
It was a massive success.
Let's quickly recap that final trap. 1, CT-179 kills OPC-like cells. 2, Depatux-M hunts down the surviving AC-like cells. And 3, together they drastically increase survival in the models.
But, we have to bring a necessary dose of reality here. scFOCAL isn't magic. There are limitations.
Right. Let's talk about those.
The most obvious one is its absolute reliance on the LANCS L1000-pound database. While they do have a shiny app interface for users to upload custom data, the core predictive power is still gated by pre-existing libraries. If a novel compound hasn't been profiled, scFOCAL can't calculate its discordance.
And there's also a major physiological limitation, right? By taking apart the tumor tissue to perform the single-cell sequencing, you completely lose the physical spatial architecture of the tumor.
That is a critical point. Knowing the proportions of the cell states is vital, but knowing where they are physically located is just as important.
Like, are the MES-like cells aggressively pushing into the healthy cortex?
Right, or are the progenitor cells hiding in the hypoxic core where drug delivery is compromised? scFOCAL is currently blind to that spatial context.
Plus, we're only looking at transcriptomics, the RNA. It misses the epigenetic landscape entirely. Factors like DNA methylation.
Exactly. Epigenetics act as the physical locks on the DNA. If we don't integrate epigenetic data or protein levels, our models have an incomplete picture. Future iterations will need to evolve into multi-omics platforms.
But even with those limitations, synthesizing the journey we've explored today is incredible. We've gone from looking at a tumor as this unbeatable shape-shifting monolith to mapping its exact transformations and mathematically calculating its vulnerabilities.
We turned its greatest evolutionary weapon, its plasticity, into a predictable trap. We are dictating its next move.
Which is just a staggering shift. So I want to turn directly to you, the listener, with a final provocative thought to ponder. If scFOCAL can mathematically predict how to force a glioblastoma cell into a specific state just so we can destroy it with a second drug, could we eventually use this exact same framework to hack cellular identity in other diseases?
That is the ultimate question.
Could we force degenerating neurons or diseased cardiac cells to revert back to healthy states entirely? We wouldn't just be treating symptoms, we would be rewriting the code to prevent failure entirely. Think about that as you go about your day.